Hello and welcome to the webinar. This webinar will cover what glycoRNAs are, glycoRNA functions and diseases, unprecedented scientific opportunities in glycoRNAs, how to profile glycoRNAs for all sample types, overcoming glycoRNA sequencing limitations, and glycoRNA research roadmap ahead. In the past, only proteins and lipids are known to be modified with sugars. RNAs are not known to be glycosylated. Defying this old concept, glycoRNAs have recently been discovered as sugar-modified RNAs. GlycoRNAs are primarily small non-coding RNAs, including miRNAs, snRNAs, rRNAs, snoRNAs, tRNAs, and Y-RNAs. So far, mRNA glycosylation has not been observed. Just like glycoproteins that can have sugars added to them via nitrogen or oxygen links, glycoRNAs can be linked via N or O links. Biochemically, glycoRNA sugars are highly sialylated and fucosylated. Due to glycosylation, glycoRNAs are located mostly on cell membrane surface, very different from their unmodified small RNA locations, such as cytosol, nucleus, or ribosomes. Therefore, they are endowed with new biological and molecular functions fundamentally different from their unmodified counterparts. For example, sialylated glycoRNAs are ligands for sialic acid-binding immunoglobulin lectin type receptors, Siglec. GlycoRNAs were first discovered by Flynn and Bertozzi in Nobel laureate Bertozzi lab. The cultured cells were metabolically incorporated with glycan mannose precursor AC4-MAN-NAC. The 4 acetyl groups allow the chemical to permeate into the cells. The total RNAs were extracted by trizol reagent method and conjugated between the N3 azide group with the alkyne bond in the DBCO-PEG4 biotin label by the Nobel Prize-winning click chemistry. The glycosylated RNAs pulled down by biotin turned out to be tRNA, Y-RNA, snoRNA, snRNA, and rRNAs. The biochemical nature of the glycoRNAs were further characterized. The glycan and RNA in glycoRNAs are covalently bonded, as stringent denaturing condition of boiling and trizol extraction could not separate them. Both N and O-linked glycoRNAs are observed, as determined by glycolytic enzymatic analysis. RNA and protein glycosylation share common glycosylation machinery. However, RNAs are not detectable in endoplasmic reticulum, a major site of protein glycosylation. RNAs do not have nucleobases similar to asparagine or serine or threonine groups for direct N or O-link formation, so the precise chemical bonding and the subcellular location of RNA glycosylation are yet to be determined. Most recently, the nature of tRNA glycosylation linkage has been determined. That is, tRNA is modified by tRNA uridine aminocarboxypropyltransferase, DTWD2, at U to form a modified base ACP3U. The ACP3U amide-modified tRNA is translocated into ER and n-glycosylated. The glycan is further matured to sialylated glyco-tRNA and presented on the cell surface. Using another labeling chemistry, the glycan is oxidized by galactose oxidase, GAO. The hydroxyl group OH is oxidized into aldehyde group and conjugated with hydrazide group immobilized on the magnetic beads. The captured glycoRNAs are released from the beads by glycosidase F, PNGase F, for N-linked glycoRNAs, or acetyl-galactosaminidase, galinase CEXO, or O-glycosidase for O-linked glycoRNAs. Sequencing analysis identified the glycoRNAs made of tRNA, rRNA, miRNA, snRNA, snoRNA, and Y-RNA. The N and O-linked glycoRNAs identified by hydrazide chemistry are small RNAs including rRNAs, tRNAs, and miRNAs. The top glycosylation site sequence motifs are predicted by PONGL-YcRNA software. GlycoRNAs have molecular, pharmaceutical, and disease functions fundamentally different from their un-glycosylated counterparts. Due to the sugar addition, GlycoRNAs are localized on the cell membrane with the glycan pointing out to the extracellular space. This changes the RNA functions fundamentally different from the canonical functions of their un-glycosylated RNAs. For example, GlycoRNAs are no longer expected to function in ribosome, or glyco-tRNAs are no longer expected to function as codon decoder in protein translation, at least directly. Instead, cell membrane-bound GlycoRNAs have their glycans exposed on the cell surface as ligands for Siglec receptors. Siglecs are important immunologically related surface molecules on the surface of immune cells such as macrophages, monocytes, B cells, T cells, dendritic cells, granulocytes, mast cells, NK cells, and leukocytes. In particular, Siglec-11 and Siglec-14 are among the first identified Siglec receptors for GlycoRNAs. Siglecs are not the only glycoRNA binding molecules. In this paper published in Cell, GlycoRNAs on the neutrophil surface bind to p-selectin on the endothelial cell surface to facilitate neutrophil adhesion and transendothelial migration to the inflammation sites. Thus, GlycoRNAs are important factors to control neutrophil immune function. Glyco-Y-RNA is a glycoRNA class that are related to autoimmune disease and a risk factor for cardiovascular diseases. Y-RNAs represent more than 60% of circulating RNAs in extracellular vehicles. Y-RNAs bind Rho60 and La proteins to become autoantigen to elicit autoimmune disease, for example, systemic lupus erythematosus, Sjogren's disease, cardiac neonatal lupus, apoptosis, and atherosclerosis. Y-RNA glycosylation and cell surface presentation can be a mechanism by which the autoantigens are translocated to the cell surface and exposed to autoantibodies to initiate injury. Y-RNA activation of macrophages and inflammation during atherosclerosis suggests that glyco-Y-RNAs could be risk factors and biomarkers for cardiovascular diseases. GlycoRNAs play disease roles in ischemic and neurological diseases. The GlycoRNAs on the surface of neuronal cells act as a ligand for Siglec-11 receptor on the microglial cell surface to suppress IL-1 cytokine and nitric oxide production, initiating anti-inflammatory cascade after ischemic stroke. This GlycoRNA or Siglec-11 axis in protective remodeling could be explored in other neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, and multiple sclerosis. In lung, GlycoRNAs are present in the extracellular glycocalyx to modulate alveolar epithelial functions. If the GlycoRNAs are destroyed with RNase, the fluorescently labeled influenza viral particles are greatly reduced for viral infection. Alveolar epithelial transepithelial electric resistance, TER, is a measure of the electrical resistance across the alveolar epithelium of the lung. TER is a sensitive and reliable way to assess the physiological integrity and permeability of the alveolar epithelium. When the GlycoRNAs are destroyed by RNase, the TER and epithelial barrier function are significantly decreased. GlycoRNAs of different small RNA classes, for example, small nuclear RNA glyco, U1, small nuclear RNA glyco, U35A, and Y-RNA glyco Y5, are broadly associated with cancers. On breast cell surfaces, from the non-cancer MCF10A to the most metastatic MDA MB231 cell lines, the higher levels of GlycoRNAs correlate with non-cancer cell types, whereas the loss of the GlycoRNAs correspond to metastatic cancer cells. In pancreatic cancer cells, many N and O-linked glyco-miRNAs have been identified. Differentially glycosylated miRNAs are related to regulation of kinase signal transduction and cell differentiation tied to pancreatic cancer cell pathophysiology. While the biological significance of glycoRNAs is continuously emerging, the potentials of surface glycoRNAs in drug development present themselves. Among all the pharmaceutical drug targets, 60% of them are cell surface molecules, with intracellular targets at about smaller 40%. Cell surface glycoRNAs may become a significant slice of drug target molecules due to the accessibility to large molecular biologics or antibody drugs. GlycoRNA targeting naked antibodies, antibody-dependent cell-mediated cytotoxicity, ADCC, chimeric antigen receptor T-cell therapy, CAR-T, bispecific antibodies, immunoliposomes, radionuclide, antibody-drug conjugate, ADC, and immunocytokines are many strategies for developing therapies of glycoRNA-associated diseases. Most glycoRNAs are observed on the cell surface. It is also very likely the glycoRNAs may end up released into extracellular space. Secreted macromolecules are important bioactive factors. By analogy to secreted glycoproteins, almost all cytokines chemokines are glycosylated. For example, interferons, interleukins, CCL1, CCL2, CCL14. Many peptidic endocrines and hormones are glycosylated. For example, insulin, glucagon, GLP-1. Glycoprotein hormones include TSH, LH, FSH, HCG. Vast majority of FGFs are glycosylated. As signaling molecules, glycosylated ligands are required for ligand and receptor engagement. For example, PDL1, dectin. In fact, glycoRNAs are already known as ligands, for example, Siglecs, and can be screened for orphan receptors. Due to their cell surface location and likely release in biofluids, glycoRNAs can well be developed as a new molecular class of biomarkers for new disease indications, live cell surface staining and sorting, easy access in biofluids, amenable to both highly sensitive antibody to glycan and nucleic acid-based detection on RNA moieties. Therefore, glycoRNAs represent unprecedented scientific and commercial opportunities. GlycoRNA profiling is essential for the study of glycoRNAs in your research areas. To profile glycoRNAs, the glycoRNAs can be captured by glycoRNA lectin binding activity. The glycoRNAs pulled down by wheat germ agglutinin beads from the total RNA are extracted by standard trizol method. The captured glycoRNAs are 3' end labeled directly with fluorescent cysteine 3C by T4 RNA ligase. The labeled glycoRNAs are then hybridized to a glycoRNA microarray for detection. This method is simple, sensitive, without the need of living cell metabolic labeling, which can be used for any type of cells and tissues. The other method of glycoRNA capture is by living cell metabolic labeling and click chemistry conjugation as described earlier. Here, the sugar precursor AC4-MannAZ is incorporated into glycoRNAs in vivo. The trizol extracted RNAs are then conjugated to dBcobiotin via in vitro click chemistry. The glycoRNAs are pulled down by streptavidin beads, fluorescently labeled with fluorescent Cy3C by T4 RNA ligase and hybridized with a glycoRNA microarray for detection. This method does require living cell culture for the metabolic labeling to work. Yet another glycoRNA capture method is by periodate oxidation of the hydroxyl group of the RNA ribose to aldehyde and conjugate the aldehyde group with the hydrazide group in the biotin hydrazide compound. The captured glycoRNAs are fluorescently labeled and detected on a glycoRNA microarray. The captured glycoRNAs can be detected on glycoRNA microarray, which covers comprehensive glycoRNA classes on one chip, overcomes many sequencing limitations, highly sensitive and accurate, requires low sample amount and quality. Technically, the captured glycoRNAs are directly end-labeled by simple Cy3C ligation, without the problems of RNA modifications and RNA folding that hinder and block reverse transcription as required in RNA sequencing library construction. PCR amplification, which can introduce bias and distortion greatly, is no longer needed. Now all classes of glycoRNAs, particularly the heavily modified tRNAs and tsRNAs, can be profiled simultaneously on one array with unbiased, accurate, and sensitive quantification, effectively overcoming the limitations of sequencing. The GlycoRNA microarray probes have terminal hairpin feature to enforce precise hybridization of small RNA terminal. Longer RNA hybridization is destabilized. GC clamp at the Cy3C of the N-labeled small RNA is designed to improve hybridization stability and specificity. The stilt spacer avoids hybridization hindrance with the glass slide. The GlycoRNA microarrays have been developed for human, mouse, and rat species. They cover all cataloged small RNAs in Y-RNA, miRNA, snRNA, rRNA, tRNA, snoRNA, and their derived small RNA fragment classes. Overall, the GlycoRNA microarrays have many advantages perfect for glycoRNA profiling. Comprehensive glycoRNA profiling. One array simultaneously covers all classes of glycosylated Y-RNA, pre-miRNA, tRNA, rRNA, snoRNA, snRNA, and their fragments. Discovery of novel glycoRNAs. Small RNAs previously unknown as glycoRNAs can be detected for glycosylation comprehensively on the array. Suitable for all sample types. Chosen glycoRNA capture method detects glycoRNAs for any sample types. Living or non-living cells, tissues, biofluids. High sensitivity and reliability. Direct glycoRNA end labeling bypasses challenges of RNA modification or folding and distorted PCR in small RNA sequencing. Unprecedented scientific and medical opportunities. The newly discovered glycoRNAs now join the ranks of biologically, clinically, and pharmaceutically important glycoproteins and glycolipids. Armed with glycoRNA profiling data, glycoRNAs can be further studied for biological and disease functions. The glycoRNA-bound partner proteins provide excellent indications of what functions the glycoRNAs do. The interacting proteins can either bind the glycan, for example, Siglecs, as glycan-binding proteins, GBP, or bind RNA portion as RNA-binding proteins, RBP. For example, to investigate the immunomodulatory potential of glycoRNAs, the interactions between glycoRNAs and members of the sialic acid-binding immunoglobulin-like lectin, Siglec, receptor family can be evaluated by immunological assays. By examining these interactions, scientists can gain insights into how glycoRNAs may influence immune responses and contribute to various disease processes, particularly in the context of neuroinflammation and autoimmune disorders. Conversely, glycoRNAs can be screened as ligands for many orphan receptors with unknown cognate ligands. Many cell lines genetically altered in glycan biogenesis have been established for analysis of glycoRNA biogenesis. For example, UDP-Gal4 epimerase gene mutation or CRISPR knockout can cause galactose or N-acetylgalactosamine defect in the glycoRNA glycans to analyze RNA glycosylation machinery. Small molecule drug inhibitors of the enzymes involved in glycan biosynthesis, such as tunicamycin, cafunincine, and swainsinine, are available to dissect glycoRNA N-linked glycosylation processes. Likewise, drug inhibitors are available to dissect glycoRNA O-linked glycosylation processes. A panel of endoglycosidases with specific cleavages for glycan structures, for example, sialidase, PNGase-F, endo-F2, endo-F3, endo-HF, and OD-glycosidase, can be used to dissect the glycan structure and the glycoRNA functions. Such studies have provided valuable insights into the mechanisms underlying glycoRNA formation and their potential roles in various cellular processes and disease states. As a glycoRNA research roadmap, total RNA is extracted from cells, tissues, or even biofluids. The GlycoRNAs are captured and profiled on GlycoRNA microarray. Differentially glycosylated RNAs can be validated by qPCR validation. The GlycoRNAs can be further functionally studied by observing the phenotypes in glycosylation gene knockout or overexpression. Blocking glycosylation steps using drug inhibitors. The molecular mechanisms can be studied by glycoRNA staining on the cell surface, by computational glycosylation site prediction, by proteins that bind to GlycoRNAs, and LC-MS analysis of glycan composition. GlycoRNAs can also be studied for their disease association and biomarker applications. I hope the information so far is helpful for your GlycoRNA research. Thank you for your interest. Please visit us at arraysdar.com.